The broadcast is now starting. All attendees are in listen-only mode.
Hello, everybody. I'm Donal Murphy, Marketing Programs Manager at PacBio, and I want to welcome everybody to the PacBio webinar series. The PacBio webinar series is a year-round offering that showcases cutting-edge research that takes advantage of the power of smart sequencing and provides insights and unique perspectives from top scientists, researchers, developers, and our industry ecosystem partners. It's my pleasure today to introduce the speakers who will present in the following order. Ben Auch, research scientist at the University of Minnesota's Genomics Center's Innovation Lab, followed by Cody Sheik, assistant professor of biology at the University of Minnesota Duluth, and finally Harm van Bakel, assistant professor of genetics and genomic sciences at the Icahn School of Medicine at Mount Sinai. We have a lot of great material to cover today, so time permitting, the presentation portion of the webinar will be followed by a questions and answers session. You are welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee dashboard. We will be recording this webinar and making it available for download in the next few days, so please keep an eye out for a follow-up email with a link to the recording. Today, our topics include details of the newly updated microbial whole genome sequencing pipeline, new insights into the ecophysiology of Minnesota microbes using long-read sequencing, and finally, understanding how whole genome sequencing is used in pathogen surveillance programs at hospitals. So with that, we'll get started, and welcome, Ben.
Well, thank you, Donal, and thank you for inviting me to present today. I'll apologize in advance if I'm a little stuffy. I came down with a cold recently. But it's a treat to be here and share some of our experiences with PacBio sequencing, both as a certified service provider and as part of our efforts in the Genomics Center's Innovation Lab. The Genomics Center is a large core facility here at the University of Minnesota. Our head count is around 40 people right now, and two of us, myself and Daryl Gohl, are part of what we call the Innovation Lab. And so we're a research group embedded within the Genomics Center whose mission is to develop novel tools and technologies for genomics research. Our goal is to improve the accuracy and expand the scope, scale, and resolution of genomic measurements. So among our areas of focus is the improvement of microbiome measurements through the use of molecular and other process standards. And today I'll present an application of our work for which PacBio sequencing has been particularly helpful, and I'll walk through the process of extraction, library prep, and sequencing of an assemblage of microbes, which we hope will have some utility for microbiome analysis. As interest in microbiome science has continued to grow, increasing attention has rightly, I would say, been drawn to the complexities of accurately measuring the composition of microbial communities. The process of generating and analyzing that data involves a number of steps, each introducing their own sets of error and bias. And so in an effort to examine these variables, a number of tools have been developed. Those include mock communities of DNA or whole microbial cells from the Human Genome Project or Human Microbiome Project, vendors like Zymo, ATCC, and others. And while these are certainly useful for studying the process in general, they contain DNA or microbes which are expected to be found in human microbiome samples. And so they can't be directly added to a sample in order to study the complete sample matrix. And they can't control for process errors or biases affecting individual samples. In response to this, several synthetic standards have also been developed, which can be distinguished from natural sequences, and thus can be spiked directly into a sample. However, these samples being only synthetic molecular constructs don't account for a critical aspect of the process, which is DNA extraction. And indeed, it's been shown that extraction remains a significant source of bias. One 2017 Nature Biotech paper focused on evaluating and optimizing the process of DNA extraction by comparing the extraction protocols of 21 different laboratories on 2 human gut microbiome samples. So you can see those 21 different extraction protocols along the bottom and different species along the left here. And they're showing the relative abundance of those various bacterial species compared to the mean. And what they're showing here is significant variability in the representation of species by these different protocols. And in particular, gram-positive bacteria remain challenging and are heavily underestimated in abundance in many of these methods. So there's still a lot of work to be done here. And while the authors use this data to recommend a few protocols which offer less bias, we think it'd be helpful to have a tool to assess this bias consistently across labs and samples, and that could potentially be used as an inline process control to track bias across the entire microbiome workflow, including extraction. And so, given this, we've partnered with Microbiologics, which is a company based here in Minnesota, which provides standards for microbiology quality control testing. We've partnered with them to develop a cell-based microbial spike-in control made up of organisms not found in the human microbiome. We refer to it as a xenobiologic microbial standard, and one prototype of this control contains an even mixture of 12 organisms. Half of them are gram-positive and half gram-negative. They span a range of GC content and genome size, as well as their source environment. And so what we hope is that we'll be able to use this tool to capture the diversity of microbial properties that might be present in a microbiome sample and track how those properties influence the resulting data. In addition, because we have enumerated cell counts on the members of this spike-in control, we expect it'll be possible to use this as a tool for calculating absolute microbial abundance. Now, in order to use our community as a control, we need to be sure that the organisms in this assemblage are well characterized. And while 16S sequences and reference genomes for these species or very close relatives exist in public databases, they're not necessarily for the exact strain we've used in our assemblage. And in the case of reference genomes, many of them are highly fragmented because they're built from short-read data. And so, in addition to all of that, we're also concerned that potentially the 16S copy number that's estimated in available databases could be incorrect, partially due to this genome fragmentation. In addition, shotgun metagenomic sequencing is increasingly being employed to replace or augment 16S sequencing. And 16S typically offers only genus-level taxonomic resolution. And because it relies on primer-based amplification of the target region, it can introduce further bias. So shotgun sequencing, even to shallow depth, can sometimes provide superior taxonomic resolution and even provide some functional information. But with either protocol, 16S or shotgun, having complete genome references specific to the members of our assemblage is going to allow us to unambiguously map reads from that shotgun data or have a high confidence in how to correct for the 16S copy number. And so, given all this, we decided to use PacBio sequencing to obtain our reference genomes. And in addition to the practical purposes, as a service provider, we were excited to use the diversity of these microbes as a sort of test bed for this new microbial multiplexing protocol, or what was then a newer protocol. And so PacBio provided some library prep and sequencing reagents to us for this study as part of a co-marketing partnership. Extraction is critical for long-read sequencing, and because the quality and size requirements are especially sensitive, the choice of extraction method can be difficult. And so in this case, I had isolates from all of these organisms as well as the community. And so in this case, I extracted the isolates using the Icahn genomic tip system, which is a gravity-based column method that uses chemical and enzymatic lysis. It typically results in really clean DNA, which is suitable for this protocol downstream. And as you can see here, the yields for our microbes are somewhat variable, even taking into account different cell inputs. But this is to be expected, and actually part of the reason we think this community will offer some utility as a tool to study extraction biases. And it's quite easy to tell visually that the lysis was not complete for many of these gram-positives. So, if you're working with isolates, that visual check can be a great indicator. One method we tried, which can assist in the lysis process and potentially improve your extraction efficiency, is the use of light bead beating. So if you're stuck, you might want to give that a try. The size of the DNA product from these extractions need to be above around 40 kb for this multiplexing protocol. This protocol is much more forgiving than larger insert protocols for PacBio, and it's certainly more forgiving than, say, nanopore might be if you're going whale hunting for super long reads. But we do need it to be above about 40 kb, not because we expect to construct libraries of this size, but because we need to be able to get a consistent shear. And starting from DNA that's already fragmented results in over-sheared libraries. So these electropherogram plots from the tape station genomic assay show that the resulting DNA looks to be primarily above 40 kb, although due to compression, that's not always super reliable. So the increased output of the PacBio sequel compared to the RS2 has really necessitated a move to multiplexing samples in order to be cost effective, especially when sequencing small genomes. And the microbial multiplexing protocol, I would say, has been improved significantly since the launch of the sequel. So I now consider it to be quite mature. Additional updates have just been published that take advantage of the recently released Express Template Prep Kit 2, but I'll be discussing my experiences with the previous protocol since that just came out. Compared to preparing libraries individually, this protocol is highly streamlined. Genomic DNA requirements are around a microgram for each microbe, and although I'd recommend starting with a bit more to account for that, sample loss that's possible, but samples are first sheared to a consistent size of around 10 kb using Covaris G tubes, and then they're cleaned up and thoroughly QC'd based on size and concentration. At this point, the library prep is individual, and the samples enter into a fairly typical PacBio library prep through the ligation step. And then barcoded adapters are substituted in the ligation step, after which point the ligase is inactivated and samples are pooled based on a calculator that PacBio provides. And that calculator decides how much of the ligation reaction should be pooled based on the concentration, the shear size, and the estimated genome size of all the samples. At this point, all the libraries are treated like a single sample, and they're moved into size selection, which is an optional step in the process, but I would highly recommend it. So this produces a fairly tight library distribution with size selection. You get a library that ranges from about 7 to 10 kb, and that's really well positioned to take advantage of the PacBio CCS reads that are becoming even more powerful with the latest chemistry. So once you've sequenced the library, then it's demultiplexed in SmartLink, and it's ready for your downstream assembly process. So you can generally plan to sequence 6 to 8 typical microbes on each SMRT cell, or a total genome content of between 30 and 40 megabases. For smaller and less repetitive genomes, you might be able to get as many as 16 libraries on a single SMRT cell, and we've become rather adept at pushing those limits here at the Genomics Center. So looking at some data from my experiences, I prepared 3 libraries with different mixtures of microbes and input amounts, and although I'll only discuss the results of one of these here, this data is from 2.1 chemistry, and it uses a 120-minute pre-extension to allow for the polymerase to get a little head start with the lights off. And this improves consensus sequencing, but more importantly, it helps increase the number of reads that can be demultiplexed and therefore used for the assembly process. And as you can see from the sequencing metrics we obtained on our sequel, the metrics are pretty consistent between libraries, and we're seeing outputs of about 10 gigabases on the 2.1 chemistry. So after sequencing, we performed genome assembly using demultiplex data from the run, and in this case, this slide, these assemblies are all constructed from the data of a single 2.1 SMRT cell, and there's approximately 32 megabases of genome content in here. So we used HGAP4, which is built into SmartLink, and we also compared this to Canoe, which is another popular long-read assembler. And from a pure contiguity standpoint, you can see that we're often getting closed genomes from both HGAP and CANU, but sometimes one is performing better than the other. And that's just based on the algorithms that they use to construct these. And so between the two assemblers, we have either one or two contig assemblies for all the organisms I'm showing here in this 7-plex. And for many of the multi-contig assemblies, there's a few that could have plasmid content, like Bordetella. I think it looks like it could be a plasmid there. And although it's hard to tell that based on coverage, but there's also the possibility that a couple of these might be what we call class 3 microbes with large repetitive regions. But considering the diversity of these microbes, it's, I think, quite impressive to be able to get 7 or more complete genomes out of a single prep and a single SMART cell. And we're continuing to sequence through the remainder of the genomes in our 12-member spiking community, and we expect to add another dozen for a second community that we're developing. We'll also plan to upload all of these assemblies to NCBI, and in addition, with PacBio, there's a bit of extra value. Alongside all of the sequencing data, there's information about methylation in the raw reads. And just a reminder that these methylation motifs can be submitted to Rebase. So I'd encourage you all to consider that with your next PacBio microbial assembly. And since I don't want to disappoint anybody, I do have some 3.0 chemistry data to share as well, which is hot off the instrument. Now, I want to emphasize that while the polymerase read lengths in 3.0 are certainly much longer, and the total base output is thus going to be greater, this isn't necessarily going to allow for higher multiplexing. So the amount of physical coverage, which does not benefit from multiple polymerase passes, has not increased because it's limited by the number of ZMWs and unique fragments. So Sequel 3.0 chemistry is going to provide an increase in base accuracy, and I think that's certainly going to help assembly accuracy. But it'll probably take a larger chip, the 8M Sequel 2 chip, for example, to allow for additional microbes to be multiplexed together using this protocol. That said, I'd anticipate that further library prep refinements might increase the shear size and the size selection cutoffs that we're using, which would increase the size of those molecules that are sequenced and offer you a little bit more physical coverage. But we'll have to see what PacBio comes up with that, and we might explore some of those ideas on our own. And you can see that with pre-extension, and this is actually a 20-hour movie, we're getting about 25 gigabases of total output on these types of libraries, which is pretty crazy. This was a lower plexity library with only 4 microbes, but I just finished the assemblies in HGAP, and they all closed up nicely. So props to Matt Areola and our PacBio team for a job well done on these. Finally, I'm very excited to announce that although we've been offering microbial multiplexing as a certified service provider for a while now, we're releasing some new aggressive pricing for PacBio sequencing through the University of Minnesota Genomics Center. We've broken it down into a few tiers to accommodate a variety of needs. So our lowest tier is simply library prep, sequencing, and demultiplexing. But for those that need to keep project costs as low as possible, then if you step that up, you can upgrade to the next tier, and we'll perform the assembly process for you, as well as run them through an automated annotation pipeline so that you can get started on your downstream analysis right away. And finally, we're offering a tier 3 service, which bundles PacBio sequencing with Illumina data to polish away the small number of indel errors that might still be present in your assembly. So this includes Illumina library prep, MiSeq sequencing, and bioinformatics. And to any of these tiers, you can also add on high molecular weight extraction or further custom bioinformatics as needed for a reasonable rate. And then for anybody in the University of Minnesota system who's listening, these are our external rates, so you can expect even lower costs than those you see here. So if you're interested in discussing this further, we'd love to hear from you. The best way to get in touch with us is to reach out to our team at nextgen@umn.edu. And in summary, I hope you can see that high-quality, complete microbial genomes are now very much within reach from both a technical and a cost perspective. We've shown that diverse microbes across GC content, genome size, and environment can be efficiently multiplexed on the PacBio Sequel platform, and they result in highly contiguous genomes. We look forward to using this data in the Innovation Lab to further development of our xenobiologic microbial spike-in control. So stay tuned for further updates on that. And with that, thanks to the great teams at PacBio for inviting me to talk today, to our partners at Microbiologics for helping us develop this project, and a special thanks to all my colleagues at the University of Minnesota Genomics Center for all their assistance. With that, I'll end and happily take any questions at the end of the webinar. Thanks.
Thank you, Ben. So Cody, let me give you the presenter baton.
All right. Hello, everybody. Welcome. It's great to be here and presenting a little bit about my lab and what we do using the PacBio system. My lab, we're broadly characterized, we study a lot of water systems here in Minnesota. But we're not really focused on specifically just water. We're really interested in the microorganisms, broadly speaking. And so we'll go into any environment that we possibly can. And so right now we have projects on sediment, phytoplankton. So these are organisms that live in the upper parts of the water column doing photosynthesis. And also just broadly speaking, the water column in general. There's a lot of interesting organisms hanging out in there that we've never seen before. And so my lab really wants to understand the biogeochemical roles of these microorganisms that we see in these environments. Because we can piece together a little bit of knowledge about the broader evolution of microorganisms by studying these evolution in these different environments. And so once we have identified some of these organisms, we're really interested in seeing the diversity of these lineages and also the biogeography of where these organisms actually occur on Earth, because this all gives us clues into how these organisms evolved as Earth biogeochemical cycles have evolved as well. And so we do spend a fair amount of time about thinking about the past environments, even though we're studying the present organisms there. Okay, and so I'm sure everybody has probably heard this at one time or another if you've never taken a microbiology course, but the vast majority of microorganisms that we see on Earth are uncultivated. This means we cannot grow them in pure cultures in the lab. And so the field of environmental microbiology and microbiology, more broadly speaking, has really had to rely on DNA sequencing to tell us, one, who they are, using conserved phylogenetic markers like the 16S ribosomal gene. But now we can actually, as we can sequence their genomes and more readily, really starting to get at this idea of their function and what their sort of roles are playing in the environment. So you can imagine here on the left, we have a microorganism with a genome that we're studying in the lab. We can sequence that genome pretty easily, assemble it back together, annotate the genes. But then scaling this up to an environment, right? So when I go out into sediments in Lake Superior or somewhere else, we may look like under a microscope look something like this. What we're really interested in is getting, since we can't cultivate most of these, is extracting out as much DNA as we possibly can, shotgun sequencing that DNA, and then trying to reassemble it back together using bioinformatic tools and pipelines. And so studying things more broadly at the metagenome scale. And so this field of metagenomics has really increased exponentially in the last few years, and that's just quite evident by looking just at the tree of life. So here we have 20 some odd years. We've seen this great expansion in not only bacteria and archaea, but also in the eukaryotes we've seen here. And so this field of metagenomics has really just opened up our eyes to what's actually out there as we sequence more and more. And it's also giving us a landscape view of evolution of organisms in different environments too. Okay, so my lab is really focused on studying these types of interactions and these microorganisms, but we really want to think about not just like the genome itself, but how it plays into the context of the greater environment, right? And so this is just kind of an aside just to show you scale that we're looking at, right? We have a zooplankton, we have this round disc is a diatom here in the center, and on that diatom there's a microorganism. And so if we think about the evolution of microorganisms on Earth and also eukaryotes, we know that microorganisms have been here for a long time, and we know that there's probably like a lot of co-evolutionary intricacies that we just can't see yet because we can't really cultivate a lot of these organisms, including these diatoms that you see here. And so really my lab is starting to transition off into thinking about more broadly, instead of just focusing on the microbe, but how these microbes interact with eukaryotic hosts potentially in the environment. So today it's going to be a little bit light on the data, but I'm just going to highlight where we're using and trying to incorporate PacBio sequencing to address our questions of microorganisms in the environment. And so we'll kind of move through a series of complexity questions here. And so when I first joined University of Minnesota Duluth, we had this culture we were using as a model organism. So this organism was isolated in 1979. It's a desulfid vibrio, just named G11. And so it's been used in numerous papers since its isolation because it's a very model organism for syntrophy. And so we were really interested in understanding this because we were going to use it as a lab culture, but it didn't have a genome. So all this time, it's been spinning out in the wild, it had no genome. So this was my first foray into using PacBio sequencing just to see what it would do for us. And so we were actually able to sequence this genome into a unitig, so one contig. We gap closed it. There was several indels, but I felt like that was pretty good for what we wanted to do with it and just use it as a lab reference. And so we published this in just Genome Announcements in 2017. Since then, we're moving on to this other isolate over here. This is as part of another project with my colleagues in Iowa State. This is a photoferrotroph. This is an organism that would have been very dominant in Earth's oceans around 2 billion years ago, probably. This organism is a photosynthetic organism. So you can see the green pigment here, and you see this orange band here, that's iron. So this organism takes light and converts it over and transfers that energy over into oxidizing iron. And so this is a really interesting organism on Earth today. And so we're trying to sequence this right now down at the Twin Cities UMN Genomics Center. So building up in complexity, moving from just these pure cultures that we have in the lab growing to thinking more broadly about microorganisms in the environment. We've started to actually use the PacBio sequencing platform to generate amplicons. And so thinking about short reads that we can PCR and PCR a gene, and we get a full read pass multiple times, so we get a high quality amplicon sequence that we can use to look at distribution of organisms across an environment. And so currently we're applying this to these phytoplankton communities in the Great Lakes. And so over here in the map, you can see all of our sampling sites that we have. We have colleagues that have been doing visual counts and looking at diversity visually through microscopy techniques. And so what we're really hoping to do is use the 18S gene, which is a eukaryotic phylogenetic marker, and amplify that entire gene and say, okay, what do we have in the Great Lakes at which time points? Well, what diatoms or other phytoplankton are we seeing here? And do those match up with the microscopy that my colleagues are doing? One of the nice things about the DNA is that we can upload it to GenBank, and now we can actually scale and have any researcher searching our sequences throughout the world. And so this may help broaden our impacts of when we try to understand other Great Lakes around the world, or even just any freshwater body. And so we're really just focusing on the 18S right now because the databases are much better than, say, the 28S or some of these other ones. And kind of keeping in line with that, the idea of using these amplicon-based studies, I was recently asked about thinking about invasive species. These are just common invasive species for Minnesota that we're really interested in. And so typically what would happen is that you would go out into the environment and spend a lot of time looking for these organisms manually. And so this would be very time intensive. And so a lot of places are starting to use this idea of DNA-based approaches to actually look for these invasives. But one of the problems with that is each invasive species typically is PCR'd with a very specific PCR primer set for just that organism. So you get a presence-absence of whether or not it's there. And if everybody's done PCR before, you know that sometimes it just doesn't work. And so you can get a lot of false positives and false negatives with that side-off approach. But it's also very limited, right? You're only looking at one species for that one water sample. And so we're hoping to do and start testing this summer is actually broadening this out and say, okay, let's amplify a common eukaryotic gene that everybody should have, or all of these species should have. And now we can actually do a broad assay for many invasives more rapidly than what we could do with just a PCR-based approach. More importantly, we can actually use that information and that data to provide a database. So now we have a basis to go about looking at different population structures potentially as well, as we have multiple invasions moving throughout these lakes. And so now getting into the fun part. So those are all great applications, I think, of the PacBio system. But here's where I'm getting real excited to really use the PacBio platform. And so we have two study sites, the Sudan Iron Mine, which is in northern Minnesota. You can go visit this as a Department of Natural Resources. They have tours. And then also this other stuff that everybody probably hates if you've ever gone swimming or walking around in a riverbed or something, and there's these rock snot diatoms here. So with Sudan Iron Mine, this is a recently NSF-funded project. I was just out there last week sampling these boreholes. You can see down here on the left, this is an iron-rich mine. This thing is this rock surrounding this is about 2.8 billion years old. And so you can kind of see where we have our drill holes down here, but we're really curious about how microorganisms thrive in this environment. And so we've done a little bit of Illumina sequencing. This is just, here we have different sampling sites here. The DDHs kind of go down. And this is all shaded out by abundance of reads that map to a specific genome. So here we have genomes coming down like this, and you can see the completion, redundancy, et cetera. And so I'll just zoom in here on this Methanolobus genome. So we see that it has one of the genome variants that we have is only found at one of our sampling locations, and it wasn't present at the other two. Well, this other one was really dominant at one and then kind of gradiated out a little bit. And so what we're really interested in is understanding why we see these sorts of patterns, right? These communities aren't very far apart. It's only about 50 meters apart from these boreholes. But given that you have to walk through, or they have to migrate through rock, that may actually be a very big hurdle for these organisms. And so what we're really interested in is using methylation patterns that we could provide from the PacBio sequencing to really look at population structure of these microorganisms that are hanging out and thriving in this subsurface environment. And more importantly, many of these genomes are fragmented, right? And so I think this is a big problem with Illumina sequencing, is that some of these genome bins, or genomes that we have, will have anywhere from 5 to maybe 300 contigs within them. So we're always asking our questions, or asking ourselves, what are we missing with this fragmented genome, right? And so we're really hoping to take that PacBio assembly, those bone contigs, and actually start to be able to map out these genomes much better and see if we are missing some very important functional genes there. And finally, this is also a very, very exciting part here, is thinking more about moving away from just microorganisms, but also thinking about these eukaryotes that hang out in these systems. So if we look at just RefSeq as of January 4th, you can see that bacteria really dominate the database, and all eukaryotes are kind of just to much lesser extent present in the database. And so if we think about evolution of eukaryotes, they came about in about the middle part of Earth's history, but we see all of these lineages here, and we see a lot of lineages that don't have any genome references, all right? And so my lab is really wanting to start thinking about doing is using this long-read sequencing technology to really go after some of these interesting organisms living in these sorts of communities and using metagenomics for it. It turns out that many of these diatoms can't be cultivated as well in pure culture. They're always going to have some sort of bacterial symbiont or some sort of bactericide with them. Within this rock snot by itself, you actually have multiple species of diatoms as well. And because it's this very polysaccharide-rich matrix that they live in, it's actually really hard to cell sort these and just pick individual cells out. And so it's a very complex environment, and we know that there has to be some sort of evolutionary feedbacks of why organisms like live in this sort of this snotty substance. And so what we're proposing to do here is generate some high molecular weight DNA and long scaffolds from these sorts of environments, and use the PacBio platform for that. And then also pair that with the power of Illumina, not only just to polish what the reads that we're getting out from PacBio, but also use a high-C technique where we can actually link intracellular DNA together before we sequence, right? And so this would help us generate these long draft genome maps that we can get from the PacBio and also from the short reads Illumina. And so really thinking about trying to construct reconstruct these diatom genomes, which are going to be very complex overall. And so that's sort of how we're starting to think about trying to go about this assembly of these sort of very complex eukaryotic assemblages. So with that, I'm happy to answer any questions that anybody has at the very end. And we have several funders, and it always takes a village to actually do all this research. So thank you all for listening. Appreciate it.
Thank you, Cody. So let me give you presenter status, Harm.
Great, thank you. So today I will be talking a little bit about how our lab is using PacBio sequencing to help define pathogen virulence and transmission dynamics in hospital setting. So I'll apologize ahead of time as well. I still have a little bit of a lingering cold, so I might cough a little bit once in a while. So moving forward, so the central interest of my lab is looking at hospital-onset infections, and these are infections that are really a large problem in many hospitals around the world. And if we know how to control those infections, we can hopefully reduce rates of morbidity and mortality in hospital. So the central questions that we have are: how do patients get infected in the hospital? Do they bring it in externally? Do they acquire the infection in the hospital? Then what is the role of colonization, the hospital environment, and patient-to-patient transmissions? Then the second question that we want to answer is: how do pathogens evolve, both within and between hosts, but also how do they adapt to hospital environments? So many pathogens like methicillin-resistant Staph aureus persist in hospitals over a long time, and they co-evolve with the patients. And in order to answer those questions, we use whole-genome sequencing. Now, to sort of start to tackle these problems, we established the Mount Sinai Pathogen Surveillance Program in 2014. And the goal here was to see if we could bring whole-genome sequencing closer to the clinical lab and make it part of infection prevention efforts in the hospital. So to this end, we established a large collection of clinical isolates, and to date we've collected about 36,000 isolates from 18,000 patients. We culture a subset of those, we perform DNA sequencing on them, and then we do whole genome and comparative genome analysis. And the lessons we learn from that, we try to sort of communicate back to clinicians and hopefully help for patient care. So we don't just do whole genome sequencing, we also integrate reports from electronic medical records, so we know where patients are in the hospital, we know their demographics, caregivers, encounters, medications, so we can integrate all this information together to get a better picture of how genomes and pathogens evolve. And we mainly use PacBio sequencing to do this, to map the spread of evolution of pathogens and to track nosocomial infections. So there are a couple of use cases that we are primarily interested in the lab. So one is mapping transmissions and outbreaks. The other is to identify risk variants, so gain or loss of certain virulence or resistance elements, and also to map and track emergence of new antibiotic resistance determinants. So today I'll be talking about the first two of these, and I'll start by mapping of transmissions and outbreaks and how we are using whole-genome sequencing to do that. So initially, when we started the program, PacBio sequencing was still quite expensive, and we were sort of gearing up to do this at large scale. So initially, most of the studies that we did were prompted by infection prevention. So they would bring us a case where they suspected a transmission. We would then gather all the isolates and epidemiological data. We would answer some basic questions. So do we know if the strain type is the same at the antibiotic profile? Of the strain is the same, and if there was a statistical increase in cases over time. So if we hit all of those check marks, we would proceed with whole genome sequencing to kind of get confirmation of potential transmissions and outbreak, and would provide feedback to the infection prevention units in the hospital. So one of the earlier cases that we studied was an instance of transmission through solid organ donor transplantation. So this was a case of a liver transplant where the donor had an MRSA infection. Usually that prevents the organ from being transplanted, but in this case there really wasn't any option, so they went ahead with the transplant knowing that the organ was potentially infected. And then indeed, within several days after the transplant, the recipient developed a MRSA bacteremia. So to investigate this case, we did whole genome sequencing on both the donor isolate and the recipient isolate, and we showed that they were genetically completely identical, confirming that this was indeed a transmission through a donor organ transplantation. Of course, our goal was from the beginning to kind of move beyond this reactive surveillance to much more proactive surveillance. So instead of infection prevention coming to us, can we do prospective, continuous surveillance and actually bring new cases to infection prevention that they were not aware of? So we started to do this kind of surveillance for two different pathogens. So methicillin-resistant Staph aureus, MRSA, which is a major cause of skin and soft tissue infections and bacteremia in the hospital. And our pathogen of interest was C. difficile, which causes gastrointestinal infections. So over a period of 3 to 4 years, we would sequence, usually within, let's say, 4 to 6 weeks of receiving the isolate, we would sequence the isolate genome. And over the years, we've sequenced more than 1,000 genomes. We're getting a really good closure rate on those. I'd say it's in the order of 85% of the C. diff genomes that we sequence and assemble are completely closed, and it's in the 90s to upper 90s percentage for MRSA. So we then do complete genome comparative analysis, and looking at genetic similarity between isolates, we identified 4 large potential outbreaks and about 42 potential patient-to-patient or smaller scale transmission events. So I'll show you an example of a little slice through these surveillance timelines and one outbreak that we studied in greater detail. So in this initial surveillance slice, it is covering a period of about 1.5 years. We analyzed 167 primary MRSA bacteremia. So we essentially sequenced the first isolate that we obtained from a blood infection of a particular patient. And for all of those, or almost all of those, we obtained complete closed genomes. We built a whole genome phylogeny of those, and sort of at a very broad level, you see the phylogenetic tree here on the left. The isolates that we sequenced broke down in either clonal complex 5 or clonal complex 8. The clonal complex 5 is a community genotype, so these are staph isolates that are typically circulating in the community, and patients may come into the hospital with, for example, an abscess that they picked up in the environment. CC8 is much more of a hospital genotype, so this is a strain that is associated with hospital stays and seems to be spreading in hospital settings. But I should say that the line between those two is blurring quite a lot, and there's a lot of sort of resident community genotype Staph aureus entering the hospital as well. Of course, because we have complete genomes, we're very interested in seeing, okay, what is the degree of variation between these genomes? We can look at the SNV level, which is basically what we get out of the core genome analysis. But we're much more interested in structural genome variation. So what I'm showing here is a whole genome alignment of 167 complete MRSA genomes. And this is a fairly complicated picture, but I'll go into a little bit more what I'm showing here. So if you zoom in to the top left over here, you can see that the solidly colored blocks are core genome blocks. So those are elements of the genome that are conserved and that are found all isolates. So they're prevalent and present throughout the MRSA tree. But we also see these accessory genome blocks, which are shown striped in this figure. And those blocks are present in only a subset of the genomes. Another thing that we see here are large-scale inversions. So these are essentially blocks where you see that the color pattern, which is ordered from red to purple, from left to right in the genome. You can see that there are blocks for certain genomes where the coloring is inverted, and those essentially correspond to large megabase scale inversion events. And the breakpoints of those inversion events are actually found in prophage regions. So they're highly homologous prophages that are present at multiple locations in the MRSA genome, and they can actually catalyze these large-scale megabase inversions. And this is the kind of results you wouldn't normally find in Illumina data unless you make very large make pair libraries, because these prophages are 40 kb in length. So it's really hard to get through those and to scaffold through those, and you really need long read data to be able to do that. So in the next slide over here, what I've essentially done is take all of these accessory genome blocks, so the blocks that are present in only a subset of genomes, and aggregate them all together in one image. And you can see that although MRSA is highly clonal, which means that it basically spreads throughout the world, and most MRSA isolates are to some degree clonally related to other MRSA isolates. Despite this high relation between them, there is actually a substantial amount of variation in the accessory genome, and about 9.5 to 16.8% of the genome is actually found in non-core blocks that are present in only a subset of genomes. So in addition to single nucleotide variant changes, there is actually a large variability in gain or loss of accessory genome elements. And I'll give some examples of the types of gain and loss of elements that we see here in this slide. So we found one instance where a strain that was not expressing Pantone, Valentine, and leukocytin, which is a toxin that has been associated with invasive growth for Staph aureus, where one isolate gained this toxin locus through homologous recombination with another strain that contained this toxin. And again, this is something that happens in an accessory genome element. This is a prophage, and these kind of homologous recombinations are really hard to find in Illumina data. Another example is shown here below, where there was a gain of a resistance-mediated element through the integration of a conjugated element. And this conjugated element contained a cobalt, cadmium, arsenic resistance gene, and those genes have been associated, for example, with the use of antibiotics in the poultry industry. And this could be a case of where one of those elements has actually inserted itself into a clinical strain. And it's really important to be able to track the movement of these kind of mobile elements. Of course, one of the other goals that we have is to see if we can do whole genome surveillance and track transmissions and outbreaks. So here I'm basically taking the tree on the left and splitting it out into the CC5 and CC8 clades on the left and on the right. And you can see that if we set a threshold of approximately SNP difference between any two isolates, we see that there were 4 different transmission events that we picked up here. 3 of these were smaller. They involved only 2 patients in each event. But there was also a larger cluster, which is T4 at the bottom right, which coincided with a NICU outbreak that was ongoing at the time that we were sequencing. And in addition to the 3 primary isolates that we sequenced, we received another 24 additional isolates that were collected during the outbreak. And we sequenced those as well. Now, the interesting thing is that if we take all of those isolates, and what I'm showing here is a new phylogenetic tree that only includes the USA 100 strain type, which is the variant that was responsible for the outbreak. You can see that the outbreak cluster shows up very clearly here at the bottom, so all these isolates are highly related. But what is also key is that this outbreak persisted over several months, which means that you get genetic drift during the outbreak itself. And that genetic drift gives you enough variation information to be able to distinguish subgroups within the larger outbreak. And again, it's very useful here to have complete genes so that you can sort of maximize the amount of available variation information. So what we wanted to see is, okay, can we use the different subclades that were identified during the outbreak to help us understand how this outbreak progressed over time? So we did that by starting to integrate genomics information with what we knew about the progression timeline of this outbreak. So what I'll show here is starting with the index case and sort of layering in additional information over time is to start to build the epidemiological timeline of this outbreak. So the index case is associated here, and for every patient, what you will see is a little bar in the middle, which indicates their stay, and the color of that bar indicates the unit that the patient stayed in. And then you'll see a dot or a square in the middle of that indicates the isolate that was collected, and the color of that isolate corresponds to one of the subclades that I showed in the earlier slide. So here we have the index case with first isolate collected at this position over here. So following the index case, in the next couple of weeks, there was a slow progression to other cases, and at some point after the third or fourth case was identified, this raised the alarm. There is an increase in frequency, and at this point we suspect that there is an outbreak. At that point, the staff in the unit started bi-weekly patient and environmental surveillance cultures to see if they could get a better idea of the scope of this outbreak. So this progressed, and in the first 2 weeks there wasn't much, but a couple of weeks later there was a huge increase in the number of cases that were detected. And you can see by the coloring that all these were part of the same subgroup in the outbreak. So there was a very rapid initial spread of the outbreak strain in the unit that rapidly added a lot of different patients on the same unit over time. So of course, it wasn't good news, and there was an intervention that included thermal cleaning. And what this means is that all the patients are moved off the unit. The unit is cleaned completely with bleach and whatever aggressive measure they could come up with. And then within a day or so, the patients are all moved back into the unit, and the hope is, okay, can we clear out the environmental contaminants and sort of start afresh? So the thermal cleaning was not successful, and we know that because there was a secondary transmission event that was actually precipitated by the thermal cleaning itself. So what I'm showing here in the green box is 2 patients that during the thermal cleaning were moved to the same unit. And during that time, they likely transmitted to each other. And the reason that we know that is because they share a variant pattern that is unique to their 2 isolates. So we know that they are more highly related to each other than to any other isolate that was selected. There was also a tertiary transmission event, again, sharing a set of variants amongst them that were not commonly found amongst the other isolates. So we know that this again was an isolated event that post-dated the thermal cleaning. So this precipitated then a second thermal cleaning and also a simulation, which means that the staff is essentially retrained in basic infection prevention strategies. And this eventually helped bring the outbreak to a close. You can see that there was still 1 purple isolate that was found over here, but we actually know that this patient was bacteremic or infected before. And we think that this infection actually happened prior to the second thermal cleaning. That still didn't tell us how this outbreak originated, right? So we thought this originated in a unit. Maybe it was a mother that sort of brought it in, or a family member that brought it into the unit originally, or a healthcare provider. But of course, we have this much larger background of surveillance in the hospital, and much to our surprise, we found that there was actually a high genetic relatedness between the outbreak strains we found on the neonatal ICU unit and earlier isolates that were collected from an adult unit, an adult ICU, several weeks to months prior. So this raised the question, okay, how did this outbreak spread between these different units, keeping in mind that these units were in different buildings, and there was no sharing of healthcare providers, so they had different healthcare provider teams, so that sort of ruled out 2 suspected transmission vectors. So what we knew is that these different places we identified in the NICU all shared locations. So we can identify these different subgroups within the NICU. They all shared a strong location overlap. But we didn't know how the transmission entered from these adult units into the neonatal ICU. What we then realized at that point was that there was actually ventilator sharing occurring between the adult units and the neonatal ICU units. And that prompted us to think about equipment as a potential vector of bringing these things into the ICU. So what we then did was go back and look at ventilator logs, but also equipment that is present in the hospital, and sort of figure out, okay, are there maybe events where we can link the movement of a ventilator to potential introduction of the outbreak into the neonatal ICU. And we identified about 3 different events that could have potentially brought this pathogen into the neonatal ICU. We also identified additional ventilator movements within the ICU that may have played a role in spreading the outbreak strain between the different locations, and explain how certain patients got affected that we couldn't account for just based on their location overlap. So really, by integrating very detailed genomic information plus epidemiological information and sort of tracking information within the hospital, we were able to really get a very detailed picture of how this outbreak occurred and progressed within the hospital. And then sort of as a final part of my talk, I also wanted to highlight some of the additional work that we've done in trying to identify the genomic features of the outbreak flow and whether there is anything specific to the outbreak flow that may have contributed to its persistence in the environment over such a long time. So what I'm showing here is again the phylogenetic tree of the USA 100 strain with the outbreak at the bottom. So we have the genomic background from the surveillance isolate and also top, and the outbreak strain at the bottom. And the yellow and red squares essentially indicate gene presence or absence. So what you can see is that the outbreak strain differs from hospital background in a number of locations. And again, it's not very surprising given our earlier results. A lot of this is sort of located in phage regions, prophage regions, and plasmids. So we see that the accessory genome it's highly mobile and it undergoes a lot of changes. So it's very useful to have PacBio sequencing there. But another thing that we noticed was that there was a change in the HSDS gene, which I've highlighted here on the left. And if you zoom into this locus, so the HSDS gene is part of a type 1 restriction modification system that consists of 3 genes. So there is a restriction enzyme, which is shown here on the left. There is HSDM, which is the modification enzyme. And then there is HSDS, which is the DNA specificity component of the entire complex. So it determines what motif is bound. So Staph aureus typically has 2 operons that give a modification and a specificity enzyme. And those 2 loci, they differ in their motifs. So one is TTC-CTT, and the other is TAC-TGG. And again, we've derived these motifs out of the methylation data that we got out of the PacBio genome sequencing. Now, the fascinating thing is that in the outbreak strain, what we saw there is a recombination event between HSDS and operon 1 and HSDS and operon 2, and that actually swapped out part of the protein, and it gave rise to a new motif, which is a combination of the 2 earlier motifs. So we get a new motif, ATC-CTT, and it's called genome-wide methylation changes in the outbreak strain. So we have a number of genetic variants, and we have methylation variants that separate the outbreak strain from hospital background. And what we were interested in, and sorry, I think it's, oh, okay. And what we're interested in is, okay, are these genetic changes and methylation changes, do they cause any change in the behavior of the outbreak strain? So we performed RNA-Seq analysis as sort of a readout of potential phenotype that could be associated with this. And we did this by comparing the outbreak strain to the 3 most closely related background strains that we observed in the hospital. So they share the same plasmid, but they didn't share some of the other genetic mutations, and they also didn't share the change in methylation profile. And what we see when we did this RNA-Seq comparison is we see increased expression in the outbreak strain of genes involved in persistence. So there are genes involved in the stress response, cell wall and biofilm formation. But we also see an attenuation of the accessory gene regulator gene expression. And we think that the combination of those 2 may predispose this strain to be more biofilm forming, and that could potentially explain why this strain was more persistent in the environment. So finally, just to wrap this up, I hope that I've shown here that building these large collections of clinical samples and bacterial isolates and combining that with whole genome analysis really allows you to study transmission dynamics and pathogen evolution in a hospital setting. It also should probably as no surprise that there is substantial variation that resides in the accessory genome, even in very highly clonally related MRSA isolates. And this is really an important aspect of the genome because it harbors virulence and resistance elements. And we've also shown that it can harbor variants that could potentially contribute to environmental persistence. So you can get to some of these elements with Illumina sequencing, but you can only really understand what's going on if you reconstruct complete genomes, which I think have shown that is really well possible nowadays with PacBio sequencing. And then finally, sort of doing continuous genome sequencing helps to reveal critical transmissions in the hospital. And what we are working on is essentially to try and get the gap between getting these isolates and getting genome sequencing data out to be as short as possible, so that we can detect transmission events before and intervene before they have a chance to grow out to much larger scale outbreaks of hospital-acquired infections. So with that, I'd just like to thank everybody that contributed to this study. So I'll just leave up the list of names because it's too much to go through and open things up to questions.
Thank you, Harm. Just a reminder to attendees that you can ask questions of the speakers by typing them in the questions panel on your attendee dashboard. And we do have some questions that have come through already. So the first one for you, Ben, is at the UMGC, do you have a PacBio protocol for fungal genome sequencing with or without multiplexing?
Yeah, we do, and it's essentially going to be the same protocol that we would use for, I mean, with multiplexing, we might do something very similar to this for the microbial multiplexing, or if we're running a singleplex, we might try to go for a slightly larger insert. But what it's really going to come down to in that case is the ability to get high molecular weight DNA out of your fungal sample. And if you've surmounted that challenge already, then we have lots of options and lots of things that we can do. But we have done quite a few fungal projects here at the Genomics Center, and extraction was always the trick. But we have a great extraction team here as well, so we'd be happy to work on that for you.
Great, thanks. Question for you, Harm. The question is, it's interesting to see the extent of structural variation in the MRSA strains you sequenced. Do you have a sense of how much virulence is mediated by SNPs versus structural variants?
So we haven't really quantified that yet, but what we're doing currently is integrating genomic data with phenotyping data. And sort of relating the changes in the genome to changes in phenotype, and we hope that that's going to give us a better impression of how big the contribution of the accessory genome to virulence is.
Okay, and this one back to you, Ben. What are other criteria apart from circularity do you have to verify the assembly outcome?
That's a tricky question. I think that's kind of a hard thing to assess. There's a number of tools out there, obviously, so you can use BUSCO analysis to look at the content of the genes in your assembly and kind of check that against a database of expected genes. You know, there's CheckM, and then in HGAP, you're going to get a stat that outputs that looks at the percentage of reads that are mapping back to the assembly, and you know, if that mapping percentage is fairly low, I would be concerned about the completeness of the assembly. But generally, we see that you know, you'd want to see that around 95% or even above, I think. But I'm not a bioinformatics person. It's not my main focus, and so tough question, but something to think about. And then obviously, if it's not sequenced, you know, you can't know whether it's there or not. So a plasmid that isn't sheared, for example, a small plasmid that wouldn't be sheared by the G-tube might just not be sequenced. It might not show up at all in your sequencing data. And so there's the possibility of missing those as well.
Thank you. Cody, a question for you. What limitations in classifications and data interpretation have you faced due to available database sequences, which are mostly short read?
That's always something that we worry about. So like with the 16S, we're really limited to, genus level. Like you can kind of look at subspecies classifications, but that's really tough, and so we're always looking to get more information. And with the 18S gene, that's like a huge problem because it doesn't evolve as quickly as, say, the 16S. And so classification of eukaryotes with just short reads is very difficult. And so there's a little bit of hand wavy, I think, when you're trying to accurately taxonomically identify these organisms. And so I think with eukaryotes, it's especially important to get as big a sequence as you possibly can. And even the full-length 18S is probably showing some limitation there as well. And so if we can extend that readout even further and maybe use, like, say, the back end of the 28S, we're also going to be able to get like an ITS sequence there, which is a much more quickly evolving sort of region of the genome in between those. And so, I think it's always something that we fight with. But if we can start generating more and more of these longer read sequences, that will help the taxonomy just in general, as we can start to think more about evolutionary relationships between them.
Thank you for that. Another question, actually back to you, Harm. Some people feel that focusing on good hygiene practices alone would be sufficient for infection control in hospitals, and that sequencing actually isn't needed. But your program has identified a reservoir of medical equipment that would have been missed by better hand washing alone. Have you uncovered other similar cases like that?
Yeah. So what I can say is that in almost all cases where we investigate outbreaks, the genomes usually tell us a slightly different story than what is expected based on epidemiology. So whole genome sequencing is really critical, I think, to understand the time frame of how an outbreak occurred. The other thing that it will give you is an awareness of transmissions and outbreaks that you would never find based on just epidemiology alone. Right. So if you have patients that are being transferred between wards, you can't really rely on an increase in frequency on any given ward, because you might actually see something spreading across multiple wards. And we have a couple of cases of these kind of cryptic transmissions that we wouldn't have detected if we hadn't done whole genome sequencing. So I think it's really something that needs to be integrated into general infection prevention practices in hospital.
Thank you. And another question for you, Harm, is, are there special requirements on the samples and or the library prep to sequence accessory genomes and get epigenetic results?
No. So the accessory genome is essentially the part of the genome that is more mobile, right? So usually when you do whole genome sequencing, if you do it on Illumina, you'll get read coverage across the entire genome, but you can't typically close it very well unless you include mate pair libraries. And the reason why you can't close it is because there are repetitive elements, mobile elements that are copied multiple times in the genome, and it's very hard to scaffold through those regions. And what PacBio does is it actually allows you to get a complete assembly so that you know exactly where the large repetitive elements in the genome are. So you get this as part of the standard PacBio genome sequencing, and you're just better able to look at those elements when the genome is complete. So that's you don't really need to do anything special for that.
Thank you. Well, unfortunately, we are out of time, and that's all the time that we have for questions. So, in closing, I would like to thank Ben and Cody and Harm for your time today and the terrific presentations that you shared. As already mentioned, we have recorded this webinar and we'll have it available for download in the next several days. So please keep an eye out for a follow-up email with a link to the recording. And in closing, I want to briefly mention three things. One is you can stay up to date on upcoming webinars as well as conferences and trade shows. You can meet PacBio at by visiting and bookmarking pacb.com/events. If you're a Twitter user, follow us for the latest news and announcements related to our technology, and our handle is @PacBio. And additionally, Ben's handle is @Cyberus, that's SCIBERIUS. and also the University of Minnesota Genomics Center handle is at UMN Genomics. And then Cody can be reached at Microbial Sheik, and the Icahn School of Medicine's Twitter handle is at Icahn Mount Sinai. And finally, if you have an interesting research project that could benefit from smart sequencing, I would have you consider applying for a SMART grant, a PacBio SMART grant, and win free sequencing. And you can stay up to date on all available grants and deadlines at pacb.com/SMART grant. So with that, thanks again for joining us today. Thank you to our presenters. I hope you'll join us again for a future webinar. And with that, take care and have a wonderful day. Bye-bye.